Welcome to Minukuu's Health Tutorials. In today's tutorial, we're going to have a discussion on superiority, non-inferiority, and equivalence clinical trials. Before going into the details of each of the three mentioned clinical trials, let's remind ourselves of the different phases of a clinical trial. Before a clinical trial, we have what we call the preclinical phase. In other words, phase 0, in which the drug is first studied in animals, experimental animals for that matter, which have similar metabolism as man. The first phase is what we call phase 1 and is the first time ever the drug would be introduced into man. And this phase is aimed at evaluating drug tolerance in man. In other words, the maximum tolerable dose of the drug in man. The study is usually carried out on a sample size of about 10 to 80 healthy volunteers. The second phase of a clinical trial is aimed at evaluating drug activity, in other words, drug efficacy or efficiency for that matter. And this phase is usually carried out on a sample size of about 100 to 300 volunteer patients. The third phase and the most important phase for biostatisticians and epidemiologists for that matter, or even clinical researchers, is phase 3. It's the phase that actually determines whether the drug will be authorized into the market or not. Clinical outcomes are usually compared to a controlled arm. In other words, the phase 3 of a clinical trial is actually a randomized controlled study in which patients would be randomized into a treatment arm, and those in the treatment arm would receive the new treatment or the new drug or the experimental drug, whereas patients that would be randomized into the controlled arm would receive the standard treatment or the existing treatment. But in the absence of the standard treatment, they would receive a placebo. The study is usually carried out on a sample size of about hundreds of patients if it is for cancer, but for regular diseases, thousands or even, as I just said, thousands of patients. In cancerology, we try to limit the number because there are fewer patients. The fourth phase of a clinical trial is what we call the post-marketing surveillance stage, where we try to aim at a long-term safety of the drug after it has already been authorized into the market. So as I said, the most important phase of a clinical trial, especially for us as epidemiologists, is phase 3, simply because that's where we actually work. We are going to compare the controlled arm and the treatment arm to know whether the new drug is more efficient, is not worse, or is having equivalent therapeutic effect compared to the existing drug or the standard drug. Having a fair knowledge of biostatistics is important in understanding the three different types of clinical trials. In biostatistics, we have two major types of hypothesis tests, either a unilateral or a one-tailed hypothesis test, and a bilateral or a two-tailed hypothesis test. With a one-tailed hypothesis test, we could have what we call a superiority hypothesis test, in other words, a right-tailed hypothesis test, based on the alternative hypothesis. What we should know is that all these tests, hypothesis tests, are named with respect to the alternative hypothesis, simply because that's where our research question is. Our research question, for example, is to show that treatment A is better than treatment B. So that's actually our alternative hypothesis. But due to random sampling error, since we didn't work on the entire populations, but we worked on samples, then we are obliged to have a null hypothesis against our research question, so that at the end of the day, if our research hypothesis, so that at the end of the day, if the null hypothesis is true, or if the null hypothesis is proved to be invalid, then we would accept our research hypothesis, in other words, the alternative hypothesis. Now, to talk about superiority or inferiority, right-tailed or left-tailed, that has to do with the statistical distribution of the given test, whether t-test, depending on the judgment criterion of our test or study. For example, right-tailed in a sense that excuse me, let me use the pointer. Assuming is z-test, for example, if it's superiority, that what it means is that, or right-tailed test, what it means is that based on the alternative hypothesis, as you can see, the difference is greater than 0. So if the difference is greater than 0, then it means our conclusion will be after 0 at the right hand side of the probability distribution curve of the given statistic. Now, as I said, if the alternative hypothesis, which is our research hypothesis, is this, for example, we are saying that treatment A is better than treatment B. In other words, treatment A is more efficient than treatment B. What it simply means is that the difference between the efficacy of treatment A and treatment B would be what? Greater than 0. Then the null hypothesis would be that if treatment A is not, greater than or equal to, if treatment A is not better than treatment B, then the opposite will be that treatment A is simply what? Less than or equal to treatment B. And that's our null hypothesis. In other words, the differences in efficacy between treatment A, B, and A would be less than or equal to 0. I'm gonna list all the different types of hypothesis tests that we have, and after that we will select from all these different types of hypothesis tests the 3 types of clinical trials. And we also have what we call inferiority or left-tailed hypothesis tests. As I said, the naming is always based on what? The alternative hypothesis, in other words, our research hypothesis. So the alternative hypothesis is saying that treatment A is what? Is less efficient than treatment B. In other words, the differences between treatment A in terms of efficacy and B would be less than 0. The null hypothesis would be that treatment A is either greater than or equal to treatment B in terms of efficacy. In other words, the differences between their efficacy would be what? Greater than or equal to 0. We could also have what we call the non-inferiority clinical trial. That one I wouldn't call it a hypothesis test, though it's a hypothesis test, alright, but usually in biostatistics you wouldn't hear of this. But it's a hypothesis test, and that's why I've included it. So at the end of the day, we would try to identify the 3 clinical trials from all these listed hypothesis tests, and why these choices by the pharmaceutical laboratories. So in non-inferiority hypothesis test, what we are trying to say is that treatment A is not worse than treatment B. That's to say, it's just like if I say let's assume we've gone to the market and then probably I aimed at buying a Casio watch. When we go to the market, you gave me a different brand, which isn't worse than what I expected. So I would say not bad. That's to say, it's the same concept. Here we are trying to say treatment A is not bad. That's to say treatment A is not worse than what treatment B. But what we are actually trying to say is that treatment A, we've actually accepted that treatment A in terms of maybe efficacy is less than treatment B. We've accepted that treatment A is less than treatment B, but not to some extent. So this is more or less like health economics, or in health economics, we often, let's say, when we are comparing different strategies, sometimes we have what we call opportunity cost, where we would say, okay, I would maybe let go some margin of, in other words, it's a certain threshold of what efficacy in order to get, let's say, or to purchase the new drug or the new treatment that has lesser efficacy to some extent. That's why we have to define let's say we have to predefine a threshold. So we've accepted that, okay, we're gonna agree to lose, let's say, an efficacy of maybe minus 5% to accept the new drug, which is drug A, which isn't which isn't worse, because we are saying is the what we are trying to say is that it's not worse than the existing treatment, but we would that's to say is actually we've actually agreed that the new drug is less in terms of efficacy, is less than the existing treatment, but not to some extent. In other words, the differences between the two drugs should not be greater than certain extent. That's to say, we've agreed that the new treatment is less than the existing treatment, but the differences should not be less than certain extent. In other words, the differences should be what? Greater than a particular threshold. Why would we want to make a drug that would be, why would we want to, not to make, but why would we want to, because phase 3 is actually about authorizing the drug into the market. Why would we want to authorize a drug into the market knowing that it has lesser efficacy compared to the existing treatments? Even though we know its efficacy is not worse, that's why we define the threshold to say it's not worse than the existing treatment based on this particular threshold. But why would we want to accept this drug knowing that there's already a drug that is booming? It's simply because sometimes the new drug might not be that much efficient as the existing drug. However, it has certain benefits. For example, it might be easily administered, probably it's having lesser side effects and what have you. So for this reason, it's like opportunity cost. We've agreed that, okay, we're going to lose, let's say, minus 5% of efficacy in order to, or minus 10%, it depends on the clinician. We're going to lose this particular threshold in order to accept it, or in order to authorize into the market, simply because it has other benefits, like fewer lesser side effects and what have you that we wish to enjoy. So what it means is that the differences between the existing treatment, which is treatment B, it could have been vice versa. Maybe treatment B would have been the new treatment, but I just prefer it this way. So the differences between the existing treatment and the new treatment must not be what? Less than a certain threshold. In other words, it should be greater than a certain threshold. So that's what we've written here. What the null hypothesis would be that the difference should be what? That's the opposite of our research hypothesis. It should be less than or equal to this threshold. Now let's look at the two-tailed or the bilateral hypothesis test. In the two-tailed or bilateral hypothesis test, we would start with what we call the inequality hypothesis test. In the inequality hypothesis test, as I said, the naming of any hypothesis test in biostatistics or epidemiology or clinical research has to always do with what? The alternative hypothesis. In other words, our research question. If we are trying to say the efficacy of treatment A, for example, is not equal to the efficacy of treatment B. But in this case, what we are trying to say is that treatment A could either be less than or equal to treatment B. In other words, their differences should not be equal to 0. Then the null hypothesis, would be that the two treatments have equal efficacy. Now, another important hypothesis test that we have, which is actually we usually have this in clinical essay or in a clinical trial, is what? The equivalence or the two-sided hypothesis test, where we would say treatment A, or in other words, the new treatment should not be what? Worse than the existing treatment to some extent. And at the same time, the treatment A should not also be more efficient or superior than the existing treatment, or in other words, the standard treatment to certain extent. Why would we say so? It's simply because we are interested in the fact that like the drug should be more efficient, so we wouldn't want it to be in a case where it's even less efficient, it shouldn't, the difference mustn't be greater than a certain threshold. But at the same time, we are saying this drug, in the case where the new treatment would be more efficient than the treatment B, it shouldn't also be, that difference shouldn't be greater than a certain threshold. Why would we want it not to be greater than this threshold? This is simply because, especially in anesthesiology, for example, there are some sedative drugs that could, lead to, it's not actually my field, but there are certain drugs that could, let's say after administration, if the drug is too efficient, the patient might just be in a coma forever. He might not be able to, to come back to normal. So we try to control the efficacy of the drug. So we say, okay, the drug should not be worse than the existing treatment to some threshold. So we have a minimum boundary or a minimum threshold of a particular, should fall within a particular interval for us to say that, okay, this drug has equivalent, the new drug, which is drug A, has equivalent therapeutic effect compared to the existing drug, which is drug B. So what it means is that the differences between drug A and drug B must lie in between the two given intervals. That is the threshold, the lower threshold, and the upper threshold. Then the null hypothesis would be that, okay, the differences between either the differences between or the difference between the two drugs would either be less than or equal to the lower threshold, or it would be greater than or equal to the upper threshold. As you can see, if the difference is less than the lower threshold here, then the opposite will be that this difference is greater than if this difference here is greater than, not less than, because it's not behind the less than sign. So if this difference is greater than the inferiority threshold, then what it means is that the inferiority threshold should be, in other words, the difference should be what? Less than or equal to the inferiority threshold. The difference should rather be less than or equal to what? The inferiority threshold. That's what that means. Here we are trying to say that, okay, the difference is less than the superiority threshold, then what it means is that the difference in the null hypothesis should rather be what? If it's less than here, it should rather be greater than or equal to what? The superiority, or the upper, I don't want to say inferiority threshold and superiority. Yes, of course, here is inferiority threshold, I'll try to say so. But usually we prefer to say equivalence threshold, since right now we are talking about equivalence trial. But in a sense that we are saying it shouldn't be inferior to some extent, so we could say, we could consider the lower threshold as inferiority threshold, and then the upper threshold will be superiority threshold. But the two boundaries gives us what we call the equivalence threshold. Now, in biostatistics, all these types of clinical trials are possible, be it superiority, inferiority, non-inferiority, because they are just simply, in statistics, they are just simply formulations. They are based on our study or our research question. But in clinical trials, you would realize that not all these are usually carried out. Why? In a sense that the pharmaceutical laboratory has their predefined objective for the drug that they are making. In your own opinion, would you want to, let's say, make a drug that would have a therapeutic effect or a pharmaceutical effect that would be greater than the existing drug, or you would prefer a drug, you would waste your time, let me not make it so clear, but would you waste your time anyway to make a drug that would be less than the existing treatment, assuming A is the new treatment and B is the existing treatment? Definitely not. Why waste your time making a drug, a new drug, that would be performing worse than the existing treatment? So definitely we would prefer a superiority trial to say the new drug is better than the existing treatment. In other words, the new drug I've labeled the treatment A is better than what the existing treatment or the standard treatment, which is treatment B. And you wouldn't want to carry out an inferiority trial. Now the question is, would you also want to carry out, let's say, an inequality trial where you would say, okay, treatment A is not, having equal therapeutic effect as treatment B. Now the new treatment is not having equal therapeutic effect as treatment B. In statistics, we could formulate these hypotheses because in biostatistics, at that point, we are only trying to criticize. But you as the pharmaceutical company, you wouldn't want to be so confused to say, okay, the new treatment that I'm making, it's like you don't know what you're about. It's not equal to the treatment, the new existing treatment. You should be certain to some extent that, okay, the new treatment that you are making is either not worse than the existing treatment, we would come back here, or is greater than the existing treatment. But you wouldn't want to say the new treatment that you are making is not equal to. If you say it's not equal to, then what it means is that it's either less than or it's greater than. So it's like we are confused. So we wouldn't want to carry out an inequality trial. Now, one important thing that you would realize here is that we wouldn't want to carry out a superiority trial simply because we wouldn't want to carry out an inferiority trial because we were aimed at producing a drug that would be better than the existing treatment. So we wouldn't want to, our research question would never be the null hypothesis, but it would always be the alternative hypothesis. So we want the new treatment, which is treatment A, to be better than treatment B, which is the existing treatment. But in a case where we are not able to prove this difference statistically or in epidemiology, then what we are trying to say, I don't know, if the null hypothesis is true, then we would fall into the trap of saying that, okay, the new treatment is worse than maybe the existing treatment. But we would never, ever, and ever, let's say, consider this hypothesis here as our objective, in a sense that it was never how we wouldn't want to manufacture a drug that would be doing worse than the existing treatment. And as I was saying, we wouldn't want to perform an inequality trial to say it's like we are confused. Treatment A is either less than or greater than treatment B. We should be more precise as a pharmaceutical company. With equivalent non-inferiority trial, as I explained, we are trying to say treatment A is not worse than treatment B to some extent. In other words, the differences between treatment A and then treatment B should not be less than a particular threshold. If it should not be less than a particular threshold, then what it means is that it should be greater than that threshold. In other words, we've accepted that treatment A is why should I say, is less than treatment B, but not to some extent. So if it's not to some extent, which is this, then what it means that the difference should be greater than this particular threshold. So we will definitely want to carry out an inferiority clinical trial, simply because a non-inferiority clinical trial, sorry. Why? Simply because in some cases for example, we have a drug that is doing so well in the market. However, it has a lot of side effects, and we are able to produce a drug that would have what? Almost the same therapeutic effect. In other words, it will be worse anyway than it will not be worse than the existing treatment. That's to say, it will have similar, or like, it's true that it will not have equal therapeutic effect. In other words, it will be less in actual sense. The new drug will be less than the existing treatment, but it has the other benefits that the existing treatment probably does not give us. For example, as I mentioned earlier on, is having lesser side effects, for example. Now, the last that I would ask you is, would we want to manufacture a drug that would obey, let's say, an equivalence hypothesis test or not? Definitely yes. In a sense, I've already stated that in anesthesiology, for example, we wouldn't want to have a drug that would be so powerful that when you administer to the patient after the operation or the surgical operation, the anesthetic or the sedative effect wouldn't stop. If they say the drug, its efficacy goes beyond this threshold, within the equivalence threshold, the upper boundary of the equivalence interval, if it's to go beyond this interval, and what it means that the patient will not be able to come back to normal. If it's also lesser than this threshold, then what it means is that the drug will be inferior to the existing treatment, which is the standard treatment B. So definitely we would agree to have an equivalence clinical trial. So you would realize that I listed 5 different hypothesis tests, but anyway, they came out with just 3. So that's to say, in clinical trials, you could have superiority clinical trial, and we could have also non-inferiority clinical trial, and then equivalence clinical trial. One important thing you should note is that the standard or the reference clinical trial is the superiority clinical trial. But in some cases, we may consider the non-inferiority and then the equivalence trial. I think I've already stated. In a case where the new treatment, we feel like the new treatment would not be that much booming or more, let's always say booming, would not be that much efficient compared to the existing treatment. However, it has other benefits, maybe lower cost, fewer side effects, and what have you. So we would want to carry out a non-inferiority test, or maybe we would want to also carry out an equivalence trial in a case where an equivalence trial in a case where we don't want the new treatment to be worse than the existing drug to some extent, and then it shouldn't also be too efficient or to some extent, especially in anesthesiology. So in order to test our understanding about all these hypotheses in the 3 clinical trials, superiority, non-inferiority, and equivalence trials, let's look at these graphs. Starting with superiority trial, let's assume the horizontal axis represents the difference in efficacy between the treatment arm and the control arm. And then point 0 represents the point of equality in terms of therapeutic effect. That's to say, at point 0, therapeutic arm has the same efficacy as control arm. In other words, or the standard treatment. That's to say, in a case where the standard treatment has the same therapeutic effect as the control, or let me repeat myself. In a case where the new treatment or the experimental treatment or the experimental arm has the same efficacy as the control or the standard treatment, then what it means that differences between the two treatments would be 0. Now let's assume we have what we call the superiority threshold. The superiority threshold is a predefined threshold or difference that we've assumed, that okay, based on the different molecules that we've used in the laboratory, the clinician will tell you that okay, the new drug that we are going to produce is not actual, or we've produced, it's not actually what should I say, it's actually gonna be superior in terms of efficacy compared to the control arm or the standard treatment by, let's say, 5%. So this difference that we've presumed, sorry, let me use the pointer. This difference that we've presumed would help us. It's a hypothetical difference that we've presumed in the actual, or we've deduced from in the actual population. This would only help us calculate the number of subjects to be included in the study. In other words, the sample size. But in interpreting superiority trial, this difference does not really matter. It doesn't take part. So let me remind you of the hypothesis that we just looked at. In superiority trial, the null hypothesis is that with a confidence level of, let's say, 95%, the interval, the interval of the difference between the treatment arm and the control arm should be what? Less than or equal to 0. This is the null hypothesis. And then in other words, the lower class bound or the upper class boundary of the interval should be less than or equal to 0. That's what it means. Now, for the alternative hypothesis, it will be that with a level of confidence of 95%, then the difference, the interval of the difference in efficacy between the treatment arm and then the controlled arm should be what? Greater than 0. That's to say, the lower class boundary of the interval of the difference between the treatment arm and the controlled arm at a confidence level of 95 should be what? Greater than 0. The lower class boundary should be greater than 0 for us to say the new treatment is what? Superior than the existing treatment. We've already seen this hypothesis, but I'm just trying to remind you. So let's start with this. Assuming this is the interval, what do we call it? The interval or the confidence interval of the difference between the new treatment and the standard treatment. Assuming this is how it's represented on the graph here. Would you say is demonstrating the new drug is demonstrating superiority or inferiority in your own opinion? Let's look at it. We can see that the lower class boundary and the upper class boundary, the whole of it is actually greater than 0. So it's definitely demonstrating what? Superiority. Clap for yourself, you had it right. Now let's also look at this. Would we say, assuming the new drug, the difference between the controlled arm or the existing drug and then the new drug or the new treatment is actually this the interval, the confidence interval of the difference? If it's like this, would you say the new drug is demonstrating superiority or inferiority, or both? Okay, I guess your guess might be right. It's also demonstrating what? Superiority, in a sense that, as we can see, the lower class boundary is greater than 0, and even the upper class boundary, the entire interval is beyond 0. So it's actually demonstrating superiority. Now let's look at the third example. Would we say it's demonstrating the difference between the existing treatment and then the new treatment? Would we say the new treatment, if this is the interval confidence of this difference, would we say the new treatment is demonstrating superiority or inferiority? This is not demonstrating inferiority, unfortunately. Superiority. Why? Simply because the lower class boundary of the interval confidence of the difference between the experimental arm, in other words the new treatment, and then the control arm, in other words the standard treatment, hasn't crossed the 0 threshold. Remember that the hypothesis is saying that for it to be demonstrating superiority, it has to cross just like these two. So definitely not. So superiority is not demonstrated. Now let's consider the fourth example. Assuming the interval confidence of the difference is as represented, is definitely not also demonstrating what? Superiority, simply because the upper class boundary hasn't crossed the 0 threshold. Now with the fifth one, would you say it's demonstrating superiority or not? Definitely not. Clap for yourself, you had it right. Let's move on to non-inferiority clinical trial. Assuming, as usual, the horizontal axis represents the difference in efficacy between the experimental arm, in other words the new treatment, and the control arm, in other words the existing treatment or the standard treatment, or maybe placebo. Definitely the not placebo, because in non-inferiority study we usually compare a new treatment to, let's say, an existing treatment, assuming that the new treatment has is less than the new treatment but not worse than the new treatment. That's to say, it's less than the new treatment, but the difference between the new treatment, even though it has less therapeutic effect, and should not be compared to the existing treatment, should not be less than a certain threshold. That's to say, it should be greater than that threshold. I'm just trying to remind what we said earlier on. Now let's assume 0 is the point of equality, where at point 0 what happens? There's no differences between the control arm and then the experimental arm. And then at point minus non-inferiority threshold, we could have fixed it. It depends on the clinician. We could have fixed that to be maybe -10. It depends. So assuming it's minus, I don't know the value, so I would say minus non-inferiority threshold. As I said, the null hypothesis would be that the lower class boundary of the 95% confidence interval of the drug efficacy difference should be what? Less than or equal to minus non-inferiority threshold. This is what we've already learned. It has to be what? Less than or equal to this particular threshold. Remember, the alternative hypothesis or research was that it should be greater than the minus negative or the minus non-inferiority threshold for us to say there's non-inferiority. This is the hypothesis actually. We are trying to say, okay, treatment A or the experimental treatment should be what? We have accepted the experimental treatment is what? Is lesser than the existing treatment or the control arm to some extent, but that difference between these two, in other words, the difference between the experimental arm and then the control arm should not, we've agreed, just like we've agreed, let's say before the experiment, our hypothesis is that, okay, let's assume the efficacy of treatment, the new treatment should maybe, let's say 2, and then the efficacy of the control arm or the existing treatment should be 3. We are just assuming. So what it means that if we are to say 2 minus 3, we're definitely going to get something negative. But we are trying to say, okay, that difference should not be greater than that particular negative threshold, to say we've agreed that, okay, the new treatment is less than the existing treatment. However, this particular difference is not, should be what? Should not be less than, in other words, should have not, instead of -1, it should not be what? Maybe -5. It should not be less than this particular threshold. So if you are saying it should not be less than this particular threshold, then what you are simply saying that it should be greater than what? This threshold, for us to conclude that there is non-inferiority. And then this alternative hypothesis is the opposite, so it should be less than or equal to the non-inferiority threshold. Let's start with the first example. Would you say it's demonstrating superiority or inferiority? Looking at it, you realize that the lower class boundary or the lower boundary and the upper boundary of the 95 confidence level, the interval, the confidence interval is totally beyond negative, the negative threshold, non-inferiority threshold. So what would we say? I would allow you to think a little bit. Okay, so as we can see, if the lower class boundary is what? Is greater than the non-inferiority threshold. In other words, if it's somewhere here, maybe so here might be, if you trace, that will be around the lower class boundary, or the lower boundary will be around 1 or 2. So it's definitely greater. So we can say it's demonstrating what? Non-inferiority, and we'll be right. And even plus what? Superiority. Why? Simply because at 0, so here is non-inferiority. It means from this point we've agreed that the standard treatment is less than what? The new treatment, but not, the difference should not be what? Less than this threshold. In other words, the difference should not be somewhere here, but it should be greater than what? This threshold. That's what we said. So now but at point 0, what it means is that the two treatments have equal or therapeutic effect. What it means that beyond point 0, the new treatment, and then the what should I say, the new treatment is more what? Is even superior than the existing treatment, in the sense that the difference has passed what? 0. Let me take it again. We said this is a non-inferiority threshold, so we're trying to say, okay, we have agreed that the experimental arm is worse than what? It's not worse, sorry, it's not worse than the control arm. That's to say, we've agreed that it's less than the control arm, but it's not worse than the control arm because the difference between the experimental arm and the control arm is not what less than negative non-inferiority threshold. So that's to say it should not be somewhere here. It's not less than this, so it should not be somewhere here. That's to say it should be what greater than this threshold. So as we can see, it's greater than this threshold. So if this difference is greater than this threshold, and it means at point 0 they have what equal, the control arm has equal therapeutic effect compared to the experimental arm. But after point 0, then what it means that the new treatment is actually demonstrating what? Non-inferiority, in a sense that is greater than the non-inferiority threshold, but at the same time it's also at greater than 0. You can see clearly that the interval confidence, or the confidence interval of the what we call it, the experimental arm or the new treatment, does not contain what? 0. So what it means that it's beyond 0 and it's demonstrating at the same time superiority. Let's move ahead. Looking at the second example, it's also demonstrating what? Non-inferiority, but this time around it's not demonstrating superiority because it hasn't passed 0. So this what do we call, the phenomenon that I just explained here is what we call the passage from what? Non-inferiority to superiority is possible. Because this hypothesis, if we had known that, let's say, the drug was actually going to demonstrate superiority, then we wouldn't have done the hypothesis test. We wouldn't have informed the epidemiologists to do anything, but we don't know. So it's a test, and it's the test results that will tell us what it is. So as we can see in the second part, it's not demonstrating superiority, but it's only demonstrating what? Non-inferiority, in a sense that it has the lower class boundary of the difference of the what do we call it, 95 confidence interval is actually what? The lower class boundary is actually greater than the negative minus, or said negative minus, the minus or the negative non-inferiority threshold. So this demonstrating what? Non-inferiority. The third example, as you can see, is also demonstrating what? Non-inferiority, but it's not demonstrating superiority because it has in the lower class boundary hasn't crossed 0. Now let's look at the fourth example. This is not demonstrating what? Non-inferiority because the lower class boundary hasn't crossed. And then the last example is not also demonstrating what? Non-inferiority. Why? Simply because even the upper boundary, everything is just outside the threshold. So clap for yourself if you got everything right. After applauding yourself, let's move now to equivalence clinical trial. So let's assume, as usual, the difference, the horizontal axis represents the difference in what? Efficacy between the experimental arm and the control arm. Then at point 0 we have the equality. That's to say at point 0 there's no difference. If I say, for example, A is equal to 5 and then B is equal to, let's say, 5. If you say A minus B is what? 0, it means there's no difference between what? Experimental arm and control arm. So at point 0 they are equal as usual. Then we fixed our lower boundary. Remember, in equivalence trial we are saying that the drug, even if at all is worse or is less than the existing treatment, that we are trying to say it's not worse. First of all, it's not worse, but we've agreed that it's less, but it should not be less than what? This particular threshold. In other words, the equivalence clinical trial is simply the bilateral, in other words, the two-tailed form of what? The non-inferiority clinical trial, if you have really observed this. The equivalence trial is a bilateral form of, in other words, a two-tailed form of the non-inferiority trial. So we have what? The superior boundary. In other words, we are saying, that's why I use the example that in anesthesiology we wouldn't want the drugs to be having so much sedative effect. So we would say they shouldn't be greater than this upper threshold. So what it means is that if the confidence interval, the lower and the upper, if everything falls within this zone, then it's this perfect equivalence. What we are trying to say is that the experimental arm has equivalent therapeutic effect compared to the existing arm based on this interval here. So let's look at this. All that I've just said, the hypotheses are here. The confidence interval of the difference should be what? For the, what should I say? Should be between minus non-equivalence threshold and then positive equivalence threshold. That's it. In the null hypothesis, either the difference would be what? If we are saying here it should be less than, it should be greater than this, then it means the difference should be less than or equal to the negative threshold. On the other way around, if we are saying it should be less than this, then what it means is that the opposite for the null hypothesis, it should be greater than or equal to the upper equivalence threshold. So without wasting my time, let's take the first example. Would we say is demonstrating inferiority, superiority, or what? What is it demonstrating? Or it's actually demonstrating equivalence. Which one? As you can see, the lower class boundary and the upper class boundary, everything is what? Beyond the positive equivalence threshold. Let's look at our hypothesis. When do we say it's demonstrating superiority? For it to demonstrate superiority, what happens? This is actually the difference. This is the difference here, but in the confidence level, that's why we have written. Why do we have even confidence level? We cannot, this is because we've done our studies on a sample, so we have to calculate within this interval. It means in the actual population, this interval contains the actual value of the difference between the two drugs. That's what it means. This particular point here represents, let's say, the mean difference. This is the exact value, but the value could be found. Let's say this is the mean value of the different possible values that could take the difference. But so this is the mean, but it takes this interval. It could take any value. We don't actually know the exact value, but this is what we call inferential statistics. We're trying to say in the actual population between this lower interval and this what this interval actually contains at a confidence level of 95%. It should contain the true value in the population. The 5% is due to a random sampling error. I don't want to go into biostatistics, but that's it. So looking at this, would we say it's demonstrating equivalence or not? As we can see, this is what, in terms of the upper boundary, it was supposed to be what? For it to demonstrate equivalence is supposed to be less than this boundary, but we can see that it's rather what, greater than this. So if it's greater than this or equal to this, what is it demonstrating? It's not demonstrating what do we call it, equivalence. So now let's move to the second one. We're saying that, looking at the second one, you will see that it's actually less than it's actually, I don't want to confuse you. You can see clearly that it's within the interval, so it's actually demonstrating what equivalence. The third one is also demonstrating what equivalence. The fourth one, the lower class boundary hasn't crossed the non-inferiority. You could call it non-inferiority in a sense that or you could call it, let's say, since we are now dealing with equivalence, we could call it the equivalence threshold, the negative equivalence threshold. So as you can see, it hasn't crossed, so this is not demonstrating what, equivalence. The fifth one as you can see, it hasn't crossed completely, so it's not what demonstrating equivalence to. Clap for yourself if you got everything right. Thanks for watching, and please subscribe.